stem cells are, and I'm going to just repeat these stem cells, okay? We all possess stem cells. These stem cells can be of myeloid lineage. These stem cells can be of lymphoid lineage. So they are the progenitors of myeloid as well as progenitors of lymphoid cells. Only few years back, maybe about 10 years back or more, a little bit more, we understood the importance of stem cells. The stem cells can be bone marrow cells. The stem cells can be in the peripheral blood cells. The stem cells, usually they are mature stem cells. So these mature stem cells, though they originate in the myeloid and lymphoid lineage, these are lineage cells, they have a specific function to perform. Correct? The problem why we started having to understand and think about these stem cells is we require stem cells which are not mature. Stem cells which are not mature, which are not destined, which are not part of particular organs are important to us for many reasons. It has a lot of importance in therapeutic medicine and medical treatment. All our organs are made of stem cells. It's a disturbance, you know. They're made of stem cells. People thought that people who have spinal cord injury, people who have genetic disorders, people who have malignancies, people who have Alzheimer's, Parkinson's, and many other neurodegenerative disorders, people who requires muscles, bones, people who require even tooth bugs, people who need transplants, skin implants, everything is attributed to these stem cells. The question here is that when the stem cells from the bone marrow is derived, they are mature, they are specified or specific cells which already have been designated. So the question here was, how do we identify these stem cells? Now you can take out a brain cell, you can identify as a brain cell. You can take it out from any organs, you identify as a particular organ. But does this mean that a skin cell can cure neurodegenerative diseases? No, because they have already been allotted, given portfolios to perform for that particular organ. I have done quite a bit of work on this. And I did this work when nobody else did it. And you will see what has happened to my work. I just want to show you. I'm not saying that I want to promote what I did, but you can see that work which I did was taken up by scientists all across the world. Now, at the time when we were thinking, we had one thing in mind: how to cure malignancies in children and adults. How we can overcome the problems that we have, especially with adult stem cells. We know that we cannot just take out the bone marrow cells and transplant it into a leukemic patient without being identical cells from identical twins, because they are genetically different. Now, if they are genetically different, we would expect our MHS or MHC, major histocompatibility complex, chromosome 6 markers to reject those cells. Now, in cells where we require has to be completely matched. It only can come from identical twins. In certain instances, the other requirement that we have, without thinking about neurodegenerative disorders and other things, was some organ transplants, like kidney or renal transplant. For that, we also require a matched stem cells. Now these stem cells, basically they are wonderful cells. But the question here is, those adult cells, we cannot actually deviate their attention to some other organs or some other function. So if a kidney is rejected, we can always come back and retransplant the kidney again. Patient will not die. There may be certain damages, but the patient will not die. A patient may become sensitized to the first kidney which he has rejected. Hence, when the second kidney is taken by that patient, that HLA antigen should not be present on that kidney, because we also have memory and spacey, and the patient who had received this has created antibodies against the first kidney's HLA antigens. So he doesn't want to face that at all. If it does, it's a violent reaction in the sense that the patient will automatically, urgently, acutely, will reject the kidney. Now, these were the two important things that we always thought we would do it. But when we started doing the stem cells as such, those stem cells, we realized how much important it is for our human body. Many of the diseases, and I personally feel, if we had a way to acquire these stem cells, then obviously there will be no clinicians, because every firm, every medicine companies will carry in bottles of stem cells. And you will go to them and say, look, I need it for Parkinson's, I need it for Alzheimer's, I need it for a spinal cord injury, I need it for this organ, I need it for that, I need it. You will buy it, right? And you will inject it. So where's the problem with the clinicians having to go through all the histories and everything? I'm just giving an example, and medicine. But is that day going to come? I still do not know. Because we have hurdles, we have ethical issues. We can't just take any stem cell and transplant into any individual unless we know that they are matching. So the question here is where to get immature cells rather than mature cells to treat different disorders. So they started with a fetus. They said the baby which is growing in the womb, their cells, their immune system is not working because the cells are not mature. So why we can't take all the stem cells from the baby, which is maturing, from the fetus, which is in the womb, from abortus fetuses, and we take plenty of these stem cells. When they found, especially in the United States, that these stem cells were so important that we can program it, you can program it into any cells of the body. So say, for instance, if I take a stem cells and put it with the neuron cells, it will grow into a neuron cells. That's the beauty of it. That will be the tolerance that will be achieved by the body, because they were immature, they cannot be rejected, they became a part of the cell structure. In America and other countries, especially women started getting pregnant and aborting the fetuses every 3-4 months just to sell that fetus for thousands of dollars. When this came to the ethical committee and they said, no, we cannot allow that, so they made it illegal. Now the scientists felt, what can we do? What can we do? So the question here was that we have to find an alternate way. So what is the alternate way? They said, okay, fine. We can tell peripheral blood. So they took peripheral blood. But these bloods are all already programmed. So that's no good for us. So finally what they said is, let's go short of a fetus. We take the cord blood from the baby as it is born. Now you have a cord blood system running in this country as well. So some volunteers of the stem cell bank go there, take a consent from the parents that when your baby is born, please, we need to take the cord blood because you're going to throw it away anyway, the hospital, burn it. So we'll take it and we extract those stem cells which are not programmed. They are the new stem cells. So many of them agree, many of them religious, this thing, they did not agree. So the question is that taking from umbilical cord, the amount of stem cells that we get is only 2 or 3 million, where we require 200 million, 300 million. So that wasn't sufficient. So the problem still remained the same. So my test, which I did it, was I was trying to find a new set of new stem cells by taking, say, my lymphocytes and taking that lymphocyte and extracting out the DNA. So that's an empty shell. The eggs you eat, like eggs when you throw everything, it's an empty shell. And inside that empty shell, we will actually inject another DNA from the neuron cells, from the spinal cord, from the muscles, from the skin of the individual. So we will insert that healthy DNA, and this cell, which now contains a healthy DNA for the organs that you need, we grow it in the Petri dish to millions. And when we grew that into millions, they became a part of that organ that you wanted. Stem cells will make platelets, red cells, different myeloid cells, lymphocytes, you name it, organ-specific cells. So how good it would be? On the basis of what I did, it became the first in vitro fertilization which was done by Steptoe and Edwards. They came to me at the London hospital. Believe me, you have to believe me. They came to me like you would come to me and said, "Doctor, how could you do this? How could you transfer?" And what I did was, at that time, we didn't know which cells were nucleated cells. But we knew that fertilization can happen through sperms and ova. So there is this sperms which is swimming enters the ova, and it fertilizes the egg. And this DNA of the sperm has actually penetrated. So there is a haplotype difference from the father is 50% and selection of 50% from the mother. When my culture of human lymphocytes I took and I inserted sperm of a third party, very unusual. I didn't know what will happen. I found after 4 or 5 days that a sperm has entered the lymphocytes, and the lymphocyte became blast cells and produces hundreds and millions of cells of that DNA. See how nice it was? So when Edward and Steptoe came to me, asked me, "How did you do this?" I took out my Petri dish, I took out 1 or 2 cultures and showed them under the microscope. They didn't tell me why they had come. They asked me what media I used to grow this. And that growth of sperms as well as the ova, I froze it. So another freezing mechanism happened, and they did not know that. So when they came to me, and both of them got Nobel Prize, when they came to me, they were in a private clinic. So I said, what I will do is I told them, I was so stupid, I told them what can be done. Said, okay, that's an excellent idea. Instead of your lymphocytes, we'll take ova. We'll take the woman's egg. And we will put the sperms of the father who cannot conceive for some reason. And what happened in their clinic? So what happened 9 months later, they had a first test tube baby, Louis Brown. They didn't mention me. They didn't tell me what I did for them. And they took all the pride. Now, at that time, in vitro fertilization was ethically not allowed. You cannot go and do all these artificial methods outside the marital system. So, my experiments, I went upstairs, and there was a research center, our research center, which had rats, big rats. And I tried the same thing of taking the ova from the female rat and taking the sperm from extracting there from the thing, did it, and I did it in a petri dish. When I did it, I sent a paper out for publication. They rejected it, and they said, we need still more proof to provide this. In the meantime, this was not allowed. Edward and Estepto went back to his clinic, set up a small laboratory, produced the first test tube baby, and when the first test tube baby was born, it came out in world papers all over, news. First test tube baby born. Medical ethics committee couldn't do anything because the result was positive. So now, where do I stand? I stand in front of you, and I'll show you what I did. My experiments actually went even further. The moment I published this in the Lancet, I got a call from America. I got a call from National Institutes of Health, National Cancer Institute, Bethesda, to come as a Fogarty scholar. I went there as a Fogarty scholar and started identifying the T cells, the subsets of T cells, the B cells, and so on. And I got a fellowship. But what did it prove from my experiment? The proof was you can have both fertilization process from the in vitro fertilization that happened. You can create new stem cells. For any organs, there is a failure, a spinal cord injury that is happening, and so forth and so on. So when I did this, the British newspaper published an article on me, one big page. Okay? The Daily Telegraph. The BBC World Services announced. The question was, that's how I got my fellowship. Had I pursued it further, possibly maybe I would have done it. Maybe I was not from the same community as white community. I was a Muslim. They didn't promote my work as such. I wrote papers, I wrote books, I wrote papers all over. And people knew what I was doing. And I'll show you this. So apart from what I'm talking about myself, any one of you can take a bold step and do something very unique. The potential is in you. It only needs a click. If it clicks and you pursue that and it happens, then obviously you are the winner. You're on top of the world. You all have the potential to do this. I didn't have any extra potential. I was normal like you. I was a student of the London Medical School. In the night time, I was doing this for some money to support my education. So this is how the things developed. Now, if you look at stem cells, Albert Einstein said this is the most beautiful thing we can experience because it is mysterious. It is mysterious because we never knew what it is. And if it is a source of true art and science. So we know now how important these stem cells came out to be. A stem cell in the blood. These may be red cells. If you look at the blood vessels, see all these stem cells floating there. What is a stem cell? It grows from one cell into millions of other cells. If you look at this one here, where it came from, all right? It came from fertilization process. What they do this is they create an embryo, they create a fetus, and the baby has actually been fertilized with the DNA of a father. These are types of different stem cells. These are the types of pluripotent stem cells. They contain all sorts of cells, as you can see, cardiac cells, red cells, you can read it, neuron cells, so on. So the body is made of cells, and they are made of these stem cells. Now, the question here was, how does it work? How does it work means that when you have cells, one single cell, and you add a DNA of the organ that you want for the stem cells to propagate into millions, you insert the DNA in here and propagate this, and this, what happens is that these cells will grow and grow and grow. One will make muscles, you name it, whatever you can, and they will grow into those cells. Those cells can repair the body of damages. Even heart and heart muscles, you can create those stem cells to repair heart and heart muscles. So what a beautiful thing that this could be. The other point here is that you can always have a cell-based delivery system. You can have a direct delivery, either through putting it into the different organs that you like, inject these stem cells, or you put it right here. When you put it right here, the cells will float in the vascular and goes to the organs which it belongs to. You see now? It's not that some will die, of course, but they will reach home. You know these pigeons? Pigeons, flying birds. You bring them, and because they have sensors, bring them many hundreds of miles, and then again tell them to fly, they'll go back to their homes because of these sensors. Similarly, the stem cells will home in to where it belongs, to which organ it belongs. Now, having said all this, you can see how much repair can happen to the cartilage, the cardiac muscles. You can have a gene therapy, and basically you are flooding the system of any single organs that are disturbed, and you can repair it with these stem cells. So what you can do now is that because we cannot get enough fetus, because we cannot get enough cord blood, what I can do, if I cut my little skin from here or here or here, or a person has a problem, when the person is burned, you want to extract some skin from the self, autograft, you can take a little skin and do the trick. Or every Muslim or Christians or whoever or Jews are circumcised. You know circumcision? Everybody knows what a circumcision is? No, tell him in Arabic, please. Okay? Now you understand? So when you cut the skin, that skin contains millions of cells. If you mash the skin, all the cells will float out. Correct? Now what do? Because the child is circumcised at birth, or the child is circumcised after 1 year, 6 months, 2 years, I'm just giving an example, you may feel that these cells may be mature to some extent. So now you want cells only. You can take out my peripheral blood, but the best thing is you will go and do invasive procedure, take a syringe, take a needle, poke it in me, take some blood out. Why not these wasted skins in circumcision? You can create a lot of cells. Now what will you do? You will, say, take 1,000 petri dishes, culture dishes. Go under the microscope. That's what I did. Select one cell only, one skin cell. Put it in one plate. Take another one, put it in another plate. So you have plenty of plates in front of you. Now you have one skin cell. What will you do? Go into a high-powered microscope, extract the DNA out of it. So that DNA, to whom this skin belongs, has been extracted and thrown away. What is left is a shell of cell. If I need to repair my neurons, I'm just giving you, I'll take my healthy neuron cells, take out the DNA and put it in here. And this will grow into millions of neuron cells of my kind.
Doctor, it's not stem cell, it's normal cell.
Huh?
This cell you have...
We'll grow it into a normal cell.
But is it
Every cell in your body is a stem cell. Every cell.
Even the mature one?
Yes. But we can't do it with mature ones. You understand? I'm giving you an alternative method how to do it. By either having immature cells, or the other alternative is, because you're not getting enough of these cells, you yourself can do this on the skin cells. I'm giving you alternate method of creating stem cells. And see how important. And people have succeeded. People are repairing, especially those with no eyesight and things like that. They are implanting all these stem cells. People who had accidents, paraplegic or quadriplegic or whatever it is, their spinal cords, their nerves. People had heart attacks. People had other organ failures. People, you can grow skin in this laboratory. I can grow skin for you in this laboratory. I can grow heart and kidneys in this laboratory. The heart will go tick, 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 tick. It will form a strong, small, and I can grow it for you.
How you arrange the cells into tissue without tissue factor or biochemical environment in the body?
Yes, okay.
You put it in the lab. How it's arranged?
Listen, there are some soluble factors within the particular environment. If say you have, I'm just giving the idea, there are certain hormones, such as pituitary hormones, adrenal hormones, so on and so forth. And these, you can propagate those cells into that particular type of cells or organs that you want. So you have a culture media which gives nourishment. You add certain hormones for that from the site, and it grows into that particular cells. That's the importance of it. Now, yeah, tell me.
Yeah, if someone lost his vision, because he lost neurocells in his vision, can he see again from this procedure?
Absolutely. I'll show you. Okay?
When you want a neuron cells that will be inserted in the skin cell, you will insert only the DNA or the whole cell?
The whole cell. The whole cell is now the neuron cell the DNA it contains. You understand now?
No.
It's changed into the stem cells of a particular type you need, of a particular organ. It doesn't act as a kidney cell if it's a neuron cell. All right? That's where, because you took the healthy neuron cells, took out the DNA, it has a DNA of neuron cells. It doesn't have a stem cells with kidney DNA.
But the skin cell here is mature enough, they do not have the potential to turn out to be a neuron cell.
Have I just told you that you take out the DNA completely, empty it out, and take it from a healthy cell from any part of the body and insert it, and that cell will grow into that cell which you need? Isn't that what I've been telling you? So now, these are therapeutic cloning or parthenogenesis, bypass embryos, and there are many ways you can do it with the skin cells. That's the whole concept behind it. These are just pictures which you may enjoy. Look at this lady. If she gets a therapy with the stem cells, she will start walking, her muscles will build up, all right? Her bones also, if problems are happening, these are some of the things. So now what is happening is umbilical cord stem cells that I've told you, or cord tissues, bone marrow, apheresis, filtering the blood from peripheral blood. You can do whatever you like, the way you want to program it. People are doing it all over the world. People have restored people's tooth, gums, even hair, eyes, you name it. So they have succeeded, and they are doing it because when we propagate and try to grow a stem cell, we expect that to grow as that. There is some problem associated with the growth problem of these stem cells, because maybe they didn't have that particular hormones or particular secretions, or in the cultures that some of these cells do die. But they are trying and they have succeeded. So there is no question of not succeeding in what is happening there. They are basically the building blocks of human beings. So we all have stem cells. We are made of cells, okay? And there are different ways a medicine will practice and alter when these stem cells are all established. I've told you, your clinical practice will fail because this man will be carrying a lot of vials of stem cells. Which one you want? Says, I want this one. Okay, fine. And then we start doing the clinical practice that way. This is the historical review of stem cells technology. This is the one of the NIH. If you look at me here, I did it with the sperms. It came out all over the newspapers, the World Services. Then after that, 79, 88, I was still a student. French researcher reported cord stem cells transplantation with Fanconi's anemia, ADD deficiencies, which is a immunodeficient type. Vision, restored vision in 2003, spinal cord injury in 2004. It keeps on doing this. Now this is one of the half page. When I did this paper, somebody wrote an article on me, okay? And this is the one which called me to United States, because they said here, if you need a bone marrow transplant or any of the other transplants, this is the way to do it. Leukemia patients. In leukemia patients, if you produce stem cells, new stem cells, not adult stem cells, then obviously this is the way to treat all these patients. And they predicted that many of these patients will benefit world over. Yes?
Doctor, where have you done this research? Here or in London?
I did it, Omar, in UK and America, both. Okay? But let me tell you one thing. Yes?
If every cell is a mature cell, why should I take cell from another person? Why should I take from my body and then change
You don't need it, man.
DNA? Change my DNA.
Why you change the DNA? Why? See, I've said this again and again to you. My adult stem cells cannot be transplanted into another human being because we are genetically different, unless we are twins and twins exchange. So we cannot transplant adult cells into another individual because the patient will die. You understand now? Or they will reject.
As a stem cell, I will take it as a stem cell.
What is the stem cells? Our body cells are stem cells. T cells, B cells, macro, they're all stem cells. Red cells, platelets, they're all stem cells. So why you want to change this? I don't know. I don't understand.
The immature stem cells.
Aye?
The immature stem cells.
I will mature it into what cells you want. Do you want your hair to be grown? I'll do that cells. If you want your teeth to be grown, I'll do that. With a new cells, with the DNA from that particular organ of the patient.
Doctor, why in China they take the stem cell directly from the fetus? They're not-
Because in China, ethically, it's maybe allowed. Maybe outside it is. You feel that, you see, I'm sorry, I don't need to criticize this. If you look at the Americans, they even have holes in the socks and they're selling it. I'm not joking, this is truth, I was there. So even though it is banned, some women are still selling secretly their fetuses.
What's the difference between fetus and from umbilical cord or from other things? Is it
Fetus will give you millions and millions and millions and billions of cells. Umbilical cord will only give you 2 or 3 million. You understand now?
Yes.
That is not sufficient to treat a leukemic patient. All right? So now, I tell you an example here. Today when you hear in London, they have got an Anthony Nolan Foundation for Cancer Research and Transplant. That's a directory they have. One of the biggest establishment, private funded establishment in the United Kingdom. Right? This work of mine created that thing. How? Why? This patient came to Anthony Nolan, a very small place within the hospital. They had one guy who was a Samoan guy, where is it? From some, Anthony Nolan was a small child with leukemia. He needed transplant urgently, otherwise he will die. So they looked at the registry and they found in New Guinea, Samoa, a man who could actually be the donor for Anthony Nolan. When he came from New Guinea, we were the only center doing this. So when I tested him, I said he is incompatible. We cannot take his bone marrow because at that time there were no stem cells. There were only adult bone marrow. So I said, no, he's incompatible, you cannot do it. That guy went back and Anthony Nolan died. And this was the actual motivation that I had to pursue it further. What I did was I take this human lymphocytes, not ova. I inserted a sperm in there. I saw the DNA, the sperm penetrate through here and grow into a blast cells of T cells of a particular DNA which this sperm was carrying from another individual. So I said, now, if I have adult cells and I can actually insert DNA into this, I might grow that into the DNA of the sperms. This is same as what you would do in vitro fertilization when people cannot conceive. That's what they do. So when I did this, why would any paper write a big article on me? Because I was creating a new science, a new generation of stem cells, a new generation of in vitro fertilization, and so on and so forth. That's what came. That's what I did. All right? But again, if you look at the books I have written, okay, these are the pictures that I had taken. So these were written in textbooks that I contributed in it. Now, having said that, having taken no honor but satisfaction of what I did in my life, and that will remain throughout my life. I don't care for any prize, I don't care for any big, I only care that I'm here teaching you. And you have to be nice to me, okay? So now when the important thing here is, how does a stem cell work? Stem cells, when you have plasticity. You can change any cell into any cell that you want according to the need of the patient by inserting another DNA from the same patient. You can have homing. If you put it, it will home to the different organs that you have destined these stem cells, that you have designated these stem cells. You can do any types of engraftment, whether they are muscles, bone, they will actually do that. The effect is absolutely fantastic. All right? These stem cells, once they are grafted, they will be self-renewing, and self-renewing. Apoptosis will happen, cell destruction will happen, but they'll keep on growing and maintaining your healthy organs. That's the importance of it. They have a potency. The potency, okay, the potency can be described in 2T. I'm not going to give you this, so don't worry about this. This has to do with fertilization process. Now, when an oocyte and a sperms in normal in vitro fertilization happens, it fertilizes. This particular oocytes will be called a totipotent or marula, and what happens is the different cells starts fertilizing. It becomes a blastocyst. That's a human fetus started to form. Now, the inner mass of these cells will produce different cells of the circulatory system, heart, muscles, and everything, including brain and different immune cells that you look for. So one cell can produce so many different kinds of things. This is the embryo, and this was the one which has millions and billions of cells. This was a stop, you cannot do it. So we have a loss of potentially life-saving stem cells because every woman in America start getting pregnant, aborting, just to sell the fetus for thousands of dollars. Okay? This was Dolly the sheep. Again, the same thing. You know Dolly sheep? Anybody? You can create two identical twins, you can create two identical sheeps based on this technology. And that's what they did. But do we need something like me, another one? I don't think so. It's not allowed. Okay? Or like you, or any one of you. Two of you walking, one to the McDonald's, one to the Kudu. And when you look at it, oh, they are both looks alike, right? Because we have created two. The second one which you create from the first one is actually weak, because they have gone through so much of transformation. This Dolly the sheep, which you received it, the second one, identical sheep, one died of rheumatoid arthritis. So the second one developed. The child of this sheep is here, grown exactly to be the mother sheep. But this died early because of complications. Now what complications? I do not know, only Allah knows. You know, why this happened. Which means probably Allah doesn't allow us to do that. So now, the concerns were, we are not using any fetuses, no, not using any babies. And again, we always have a problem making all these stem cells. The sources of specialized cells, you can achieve it whichever way you like it, whether you take a bone marrow cells, make it into stem cells, which is adult. You can take it from the liver, the brain, or wherever it is problem. You can always make a stem cells and inject it into the humans. This is how it happens. A cell which has been fertilized. Okay? It goes through 1 to 5 days, divides again and again, and called a blastocyst. This is how the cells in the blastocyst starts looking around, and it can, these different cells which are growing within a cell has got different functions. One may be neuron cells, one may be bone cells, one may be muscle cells, one may be liver cells, one may be kidney cells. So now look at, if you leave it to mature, they will form different parts of the organ. So that's what we produce every time. You think that all these stem cells are useless in our body? No, they are still repairing our different systems. We get sick, they are still repairing it, yes.
So there is a difference between the embryo stem cells and the bone marrow stem cells, other than the numbers. Is the quality?
The quality,
Yeah.
look, less invasive procedure, less experimentation, less time, you can take an adult bone marrow. But as I told you, it has very restricted, limited function. If you do the stem cells, it does take time. It's continuously happening. People are growing stem cells. That's much more nicer because there will be no rejection. So that's the importance of this. So look at this, how the stem line of stem cells grow. They grow into different cells. Now, those stem cells, these, what they will form? They'll form pancreatic islet cells, so insulin will be defeated. We can have muscle cells, people who go through nerve cells, so all the genetically inherited diseases will be gone. Now, I am not going to go into a lot of this. You have understood this is just a part of the colony of stem cells that can be grown in a petri dish, millions of cells. You can use it. If they are neuron bond cells, you use it there. If muscles, bones, whatever, you use that. This is the cells that you take from a circumcised baby, how you can grow into a different types of cells. I'm not going to go. So now you can have Parkinson's, heart disease, diabetes, everything that these are the cells which are growing in the culture, high-powered microscope, colored, and you can see, you see the heart, you see how many things you can do there, the brain, the spinal cord, the bones, the teeth, the heart, everything you can grow with these stem cells and repair it. That's the importance of it. Look at here. This is grown in a petri dish. This is a kidney, a heart. Okay? You have red cells growing. So you can grow whatever you like. You don't need to go to a blood transfusion van and give blood for donation. We can grow it in the laboratory for you. Whatever you need, platelets, red cells, whatever you need, we will grow it for you. These are just the different part of it. I'm not going to go into adult stem cells. You already know. I know we have talked about enough of it. Okay? You can see here how they inject the heart muscle regeneration in the mouse. They injected the stem cells in the mouse. Experimentation is done on mouse, monkeys, dogs, etc., etc. This is done in the mouse. As you can see here, what they are doing, how they are injecting this. Heart muscles, these are the cells, so they will repair the heart muscles very nicely. I think I come to the end. Any question? I hope you enjoyed it. It's fascinating, even if you don't get questions out of this. But you know something which you have not known before. So, important thing is you have gained some knowledge of it. All right? Thank you. Take care.